Life and death aren't necessarily absolute states. The cellular world has its own rules. Hey there, welcome to Science is You. If you're enjoying these videos, please subscribe to help this channel get off the ground and to support our mission of making science accessible to everyone. We often envision growth as a linear path from life to death. It's true that the cells that make up your body are growing and dividing, but at the same time, cells are also dying. In fact, this delicate balance of cellular growth and death is critical for health. For example, if cells stop dying and just proliferate, this can result in overgrowths and cancer. Cellular death is necessary to remove cells that are injured or abnormal. Alternatively, if cells stop growing and don't replace the ones dying off, then eventually there wouldn't be enough cells for our organs to maintain their structure and function, and can lead to degenerative diseases. Cellular death is just as important to life as cellular growth. Just as cells have systems and mechanisms in place to enable their division, they also have specific mechanisms that cause their death. One of these, called apoptosis, is named after a Greek word that means falling to death. Apoptosis can be thought of as cell suicide. In this process, cells actually produce proteins that chew up the cell's DNA and destroy the cell from the inside out. It was previously believed that production of caspases by a cell basically was a point of no return beyond which the cell could not survive. That's logical. I mean, if a cell's DNA is all broken up, it's done, right? Well, this paper demonstrated that cells actually can reverse the apoptosis process of cellular death and recover from late stages of this death process, including DNA damage and destruction of mitochondria, which are the parts of the cell that produce energy. High levels of a toxic substance like ethanol can cause cells to begin dying via this apoptosis process. Wait, but how can you tell that the cells are dying? When cells start dying through apoptosis, they sort of shrivel up and start breaking apart with these bubble-like protrusions called blebbing. Also, the nucleus where the cell's DNA is kept can appear smaller and broken apart. Fragmenting of mitochondria that produce the cell's energy can also occur. Here, the researchers were studying a few different types of cells, specifically liver cells, heart cells, and fibroblasts, a type of skin cell. They exposed the cells to a toxic substance for several hours and observed these classic signs of apoptotic cell death. But shockingly, after the toxic substance was washed away, the cells began regenerating and recovering. By 24 hours later, over 90% of all cells had completely recovered. Videos of individual cells show how they begin to die and even begin to break apart. But after the toxic substance is removed, they somehow manage to rebuild themselves and survive. The authors named this phenomenon anastasis, which is a Greek word meaning rising to life. Let's take a closer look at what's going on inside the cell. When the apoptosis process of cellular death is triggered, proteins called caspases destroy the cell. The authors here observed that one such protein, caspase 3, was activated in the presence of toxic levels of ethanol, but after washing was deactivated. These so-called executioner caspases are present in healthy cells but held in an inactive state. Toxins or other cues can trigger a chain reaction within the cell that results in the activation of caspases. The caspase has to be cleaved or cut to function. You can sort of think of this like a vicious dog on a leash. When it's muzzled on the leash, it is controlled and can't really do anything, but if you cut the leash, it's free to attack. It was previously believed that after caspase activation, a cell is doomed to die. To further understand what is going on inside the cell, the authors genetically engineered cells to produce a caspase biosensor. To do this, they inserted a gene into the cell to produce a special protein. Proteins are long folded chains made up of smaller molecules called amino acids. This biosensor protein contained a special set of amino acid sequences. This protein contained a portion also that tethered it in the cytoplasm, the main inner portion of the cell surrounding the nucleus, a large circular area where the cellular DNA is kept. This tether was connected to a fluorescent molecule that could be taken up by the nucleus. If active caspases were present, they could cut the tether at this special amino acid sequence. The fluorescent molecule could then move into the nucleus. Living, healthy cells that have a fluorescent signal in the nucleus would then mean that they had active caspases at one time, but then recovered. For example, when the authors treated cells with a toxin for a few hours, they saw the fluorescent color move from the cytoplasm into the nucleus, along with the bleeding and other signs of cell death. After removing the toxin and washing, many cells retained the fluorescent molecule in the nucleus. This indicates that cells can indeed recover from active caspases. As mentioned earlier, during cellular death, the DNA is broken up and destroyed. The researchers detected significant DNA damage after application of toxins. How then could these cells survive? Upon a closer look, the authors discovered that surviving cells had repaired much of their DNA, but some damage remained. Specifically, they noted that many cells contained micronuclei, multiple small nuclei instead of one large central nucleus for storing DNA. This could be thought of sort of like if you had a library full of books. The books are your DNA. Then someone takes the books and throws them into lots of different boxes. The books are still there, but maybe some are damaged and the situation isn't as nice. Further, the damaged DNA raises some questions. Can DNA damage and mutations cause cancer? Exactly. The breaking apart of the cellular DNA during death and then putting it back together during recovery can cause massive genome rearrangements. The authors noted that a fraction of cells that had recovered from death displayed characteristics of cancer cells. So, recovery from cellular death could indeed be a way that cancer could form. Wait a second, even if cells could rebuild and survive after their DNA is so damaged, wouldn't that mean that their proteins are all chewed up and the cell is largely destroyed? How could it recover from that? So, unbelievably, the authors were able to detect transcription going on in the cell during recovery. Transcription is the process of making RNA from DNA. RNA is used to make proteins. The authors applied a drug that prevents the cell from making RNA. This greatly hindered the ability of the cell to recover from death. This suggests that the cell needs to be able to actively start producing new proteins in order to rebuild itself and recover. But what does this mean? Do we have zombie cells living inside of us? The authors speculated several intriguing implications from this study. For example, the phenomenon that cells can recover from late stages of the death process with massive genetic changes could help explain why repeated injury or alcohol abuse appears to increase cancer risk. More disturbingly, this could also be a means through which cancer cells survive chemotherapy and radiation treatments and become more aggressive or even acquire resistance. On the other hand, the anastasis recovery phenomenon might not be all bad. For example, this could serve as a means to preserve injured cells that are hard to replace, like neurons or heart cells. Future research will likely uncover many exciting prospects as we learn more about the incredible abilities of the cellular world. If you liked this video, please don't forget to subscribe to help this channel get off the ground. And remember,